wondered how new drugs are designed for diseases like cancer, HIV, or even rare genetic disorders? How scientists pinpoint the perfect molecule to target a deadly virus, all with the click of a button? Well, buckle up, because today we are diving into the fascinating world of molecular docking and structure-based drug design. A tool so powerful it's revolutionizing the pharmaceutical industry. This is your ultimate beginner to advanced guide, where discovery meets technology and the future of medicine unfolds. You don't need a lab coat for this one, just curiosity. If you are a bioinformatics enthusiast, a student, or someone just fascinated by the cutting edge of computational biology, this video is for you. Not only will I break down molecular docking step by step, but I'll also guide you through its real-world applications. So by the end of this video, you can understand how this cutting-edge technique is changing the way we fight diseases forever. Let's get started. What is molecular docking? Let's start with the basics. Molecular docking is like solving a three-dimensional puzzle on a molecular level. Imagine you have a lock, the protein, and you are looking for the perfect key, the drug or ligand, that fits into it. What's the goal? To figure out the best way these molecules can interact, how strong their bond is, and whether they can trigger or block a biological process. Molecular docking is a computational method to predict how a small molecule, a ligand, binds to a macromolecule, usually a protein, and evaluates the strength of their interaction. If you talk in simple terms, it's like coding for molecules. Which ligand pairs best with which protein in a way that sparks biological fireworks or shuts them down entirely. And yes, it is as exciting as it sounds. Why is molecular docking so important? Before molecular docking, drug discovery was like finding a needle in a haystack. Scientists would spend years and billions of dollars testing drugs in labs. But now, computational docking simulates these interactions virtually. This accelerates drug discovery, reduces cost, and even helps solve mysteries behind previously untreatable diseases. Did you know that drugs like HIV protease inhibitors, which help turn HIV from a death sentence into a manageable condition, were developed using molecular docking? Amazing, right? Alright, let's demystify the actual process. As promised, beginners, intermediates, and advanced learners, this part is for you. Molecular docking has 5 key steps. Let's break them down. Step 1. Prepare the protein, your target. Protein Data Bank website being accessed, downloading a PDB file. First, identify your target protein, usually a biomolecule responsible for causing or influencing a specific disease. Use tools like the Protein Data Bank to retrieve its 3D structure. Step 2. Prepare the ligand, the key to the lock. OpenPebble tool converting chemical structures into readable format. Next, prepare the ligand, the potential drug molecule. Using software like OpenPebble, you can clean, optimize, or convert it to formats like PDBQT for docking. Step 3. Grid generation. Autodock tool setting up a grid around the receptor. Here is where you decide where to dock. This is usually the active site of the protein, the part where a reaction or inhibition takes place. Tools like Autodock tools create a grid and mark this area. Step 4. Docking simulation. Autodock running simulations and generating binding poses. This is the star of the show. Using software like Autodock, the simulation evaluates thousands of possible poses of the ligand and calculates their binding energy. The lower the binding energy, the better the fit. Step 5. Analyze results. Pymol showing molecular interactions. So finally, you analyze the results. Does the molecule fit? How strong is the interaction? This helps rank potential drug candidates based on their energy scores. Now you are probably wondering, how does this translate to real life? Good question. So COVID-19 drug development. During the COVID-19 pandemic, molecular docking was used extensively to identify compounds that could block the SARS-CoV-2 virus from binding to human cells. Next, cancer therapies. Color-coded molecules showing protein inhibitors. Docking helps design drugs that can turn off mutated proteins present in cancer cells. Third, personalized medicine. In the near future, we may design drugs tailored to your unique genetic makeup thanks to molecular docking. Challenges and misconceptions. Okay, so we have talked a lot about the power of docking. But let's get real. There are challenges. This isn't just magic, it's science. Results depend on accurate protein structures, ligand availability, and experimental confirmation. A common misconception is that the docking alone creates a drug. It's false. It's just the beginning. Experimental validation in wet labs is still absolutely crucial. Now, if you are ready to dive into the world of docking, here are some must-know tools. For beginners, Autodock Tools, Discovery Studio Visualizer. These are user-friendly and great for learning how docking works. For intermediate and advanced users, Schrödinger, MOE, and Docking at Cloud for high-accuracy docking with advanced algorithms. The future of drug design lies at the intersection of molecular docking, artificial intelligence, and genomics. AI algorithms can now predict binding affinities faster and with greater accuracy than ever before. Imagine a world where diseases are fought with tailored drugs designed virtually before a lab gets involved. Whether you want to work in drug discovery, bioinformatics, or just want to understand the future of medicine, molecular docking is a skill worth mastering.